Hello everyone, we are the Group 3 of Psych 2A and we will be discussing about the experiment on lipids. The next slide contains the group members and we are composed of 12. I am Eliana Mystica and I will be first discussing lipids, its description, functions, and foods that are high in lipids. Kasi there will be terms, words, or even phrases that will be mentioned throughout this report that I believe we are not yet familiar of. So, to start with my report, first, we are all aware that lipids are fats. And lipids are organic compounds kasi binubuo siya ng hydrogen, carbon, and oxygen atoms. Also, lipids are nonpolar kasi yung ends ng lipid ay not charged. Given that they are nonpolar, they are insoluble in water kasi they do not bond with each other or they do not share electrons in any way. So, yung mangyayari kapag i-dissolve natin yung lipid sa water, magfo-float lang siya. Also, usually are synthesized in the human body. It is commonly present sa liver, and it can be found in oil, butter, whole milk, cheese, fried foods, and some red meats. Nadadigest natin yung lipids with the help of an enzyme called lipase. Yung lipase, it breaks down the lipids into fatty acids and glycerols, which is helped by the bile in the liver. Here are the functions of the lipids. First, lipid is a component of the cell membrane structure. Kasi we have mentioned that lipid is a nonpolar. So it keeps the cell membrane impermeable to water. And yung ibang water-soluble substances, mahirapan sila mag-cross sa lipid. Second, lipids acts as a storage for energy in the body. Kasi our body uses carbohydrate as a direct form of energy and it is stored sa lipids. So third, lipids allows the body to use vitamins such as vitamin A, D, E, and K, which is beneficial to the body. So last is, lipid helps you in your diet kasi lipids in your liver allows fat and water to mix sa intestine and it helps sa pag-breakdown and absorption of the nutrients. Here are some foods that are high in lipids but some are good for your body while some are not. Let us first start with the avocado. Avocado has 77% of fat calories. However, yung tinutukoy na fat dito is monosaturated, kung saan tinatawag itong healthy fats because it helps in lowering the LDL and increases HDL. Also, did you know that avocado is rich in fiber, which means na kahit mataas siya sa lipids, it is still healthy. Kasi it is a good source of fiber which is important for a healthy digestive system. To be followed by the cheese. Cheese is known to be high in saturated fats kasi kaya nitong itaas yung level of cholesterol in the blood. However, a single slice of cheese ay katumbas ng calcium na makukuha natin sa isang glass of milk. And even a gram of cheese has more protein than any other meals. So next is an egg. Many believe that egg is harmful kasi it has a high cholesterol concentration sa yolk, which means it has 212 milligrams, in which accounts 70% of the RDA. Yung RDA stands for the Recommended Dietary Allowance, kung saan yun yung daily average intake level na para ma-meet yung nutrient requirement ng isang tao. However, yung cholesterol na makikita sa yolk does not increase the blood cholesterol level ng isang tao. Kahit na mataas yung content level ng lipid, it is important in any weight reduction diet kasi they are high in proteins which helps build muscles and it will help you make you feel full. Now, let us move on sa mga foods that are high in lipids but not beneficial for you. As you can see, we have fried foods, cakes, and frozen desserts. Kaya sila nasabing not beneficial for you because they are high in lipids in which it can be saturated or trans fat. Both are rich, not healthy, and can increase the risk for a heart disease or any other complications. If these foods are not avoided, it will develop ailments and other health complications kasi they are high in lipids. So the next presenter will be discussing the test for solubility.
Hi, my name is Cheryl Luis Hernandez and I will be presenting about test for solubility. This type of test is performed to determine if a solute is able to dissolve in a liquid solvent. A solubility test is essential to establish whether a solute is acidic or basic and helps find out the polarity of the liquids involved. For more context, a brief explanation of polarity is when the molecules in the components used are positively or negatively charged. A polar solute will dissolve in a polar solvent, whereas a nonpolar solute will dissolve in a nonpolar solvent. Although, if we put a polar solute in a nonpolar solvent, it will not dissolve and vice versa. While an acidic solution has a high concentration rate of hydrogen ions, while a basic solution has a low concentration rate of hydrogen ions. The chemicals used as solutes in this experiment are coconut oil, glycerol, which is a simple polyol compound that is colorless, odorless, and thick stick liquid, which is sweet tasting and non-toxic, and palmitic acid, which is the most common saturated fatty acid found in animals, plants, and microorganisms. While the eight chemicals used as solvents are water, dilute acid, and dilute alkali, for those who are unaware, dilute means water has been added to the mixture to make it thinner or weaker. And chloroform, a chemical compound that is colorless, strong smelling, dense liquid. Ether, a colorless chemical which is sweet smelling and extremely flammable. And cold alcohol, hot alcohol, and finally, carbon tetrachloride, which is commonly used as a dry cleaning agent. Step 1. The test starts out with 8 test tubes laid out containing one solvent per tube with an equal amount of 1 milliliter. Then on step 2, choose one solute to first conduct the test, then add one drop of it on each test tube containing the solvents. Also take note if the solute immediately mixes with the solvent. Step 3. Tap the test tube with your finger to mix, or stir gently with a glass stirring rod. Shaking the test tube vigorously after the addition of each portion of solute is also optional, although I would advise you to do it with caution. As a reminder, the test tubes are fragile and may break. Repeat steps 1, 2, and 3 with each solute after observing and recording the changes. As seen on step 4, solubility will be indicated by the turbid solution or clarity of each test tube. In other words, the clearer the solution is, the more likely it is soluble. While when a negative result has been obtained, the lines where the two liquids separate are visible through the whole solution. A more in-depth explanation of the test results will be further explained by the next reporter. Thank you.
I'm Kimberly Garabat and I will discuss to you the result interpretation of the test for solubility of lipids. The goal of test for solubility is to determine whether a lipid is miscible or not in a particular solvent. If it dissolves with a specific solvent, it means the lipid is soluble with it. Note that lipids are nonpolar, which means it is only soluble in also nonpolar environments. So as you can see on the table, the topmost row entails the solvents, while the leftmost column contains the solutes. Solutes are substances that would be dissolved in another substance. In this experiment, coconut oil, glycerol, and palmitic acid serve as our solutes. On the other hand, the solvents are water, dilute acid, dilute alkali, chloroform, ether, cold alcohol, hot alcohol, and carbon tetrachloride. The negative sign and positive sign in the results serve as our legends. The negative sign denotes for insoluble, while the positive sign is for soluble. Let me tackle first coconut oil and its solubility in various solvents. First, coconut oil is not soluble in water. It would only float as coconut oil is nonpolar, whereas water is a polar solvent. Next, coconut oil is insoluble in dilute acid due to the coconut oil's higher molecular weight. With dilute alkali, coconut oil is insoluble in it. On the other hand, coconut oil can dissolve in chloroform due to both of them being nonpolar. Next, so ether is an organic nonpolar solvent, meaning it can dissolve coconut oil. Next, with both cold and hot alcohol, coconut oil also did not dissolve. And lastly, since carbon tetrachloride is also a nonpolar solvent, it means it can also dissolve coconut oil. Onto the next row, we have glycerol as our next solute. Notice how it is insoluble with all of the given solvents, This is due to the nature of the solvents, the nature of glycerol as a solute, the amount of solute, and the rate of solution. And with our final solute, we have palmitic acid and its solubility with the same solvents as the two prior solutes. Palmitic acid did not dissolve in water, dilute acid, dilute alkali, chloroform, hot alcohol, and carbon tetrachloride. However, it is soluble in ether and cold alcohol. This is because palmitic acid is highly soluble in organic solvents such as ether. I am Kimberly Garabat and I will discuss to you the result interpretation of the test for solubility of lipids. The goal of test for solubility is to determine whether a lipid is miscible or not in a particular solvent. If it dissolves with a specific solvent, it means the lipid is soluble with it. Note that lipids are nonpolar, which means it is only soluble in also nonpolar environments. So as you can see on the table, the topmost row entails the solvents, while the leftmost column contains the solutes. Solutes are substances that would be dissolved in another substance. In this experiment, coconut oil, glycerol, and palmitic acid serve as our solutes. On the other hand, the solvents are water, dilute acid, dilute alkali, chloroform, ether, cold alcohol, hot alcohol, and carbon tetrachloride. The negative sign and positive sign in the results serve as our legends. The negative sign denotes for insoluble, while the positive sign is for soluble. Let me tackle first coconut oil and its solubility in various solvents. First, coconut oil is not soluble in water. It would only float as coconut oil is nonpolar, whereas water is a polar solvent. Next, coconut oil is insoluble in dilute acid due to the coconut oil's higher molecular weight. With dilute alkali, coconut oil is insoluble in it. On the other hand, coconut oil can dissolve in chloroform due to both of them being nonpolar. Next, so ether is an organic nonpolar solvent, meaning it can dissolve coconut oil. Next, with both cold and hot alcohol, coconut oil also did not dissolve. And lastly, since carbon tetrachloride is also a nonpolar solvent, it means it can also dissolve coconut oil. Onto the next row, we have glycerol as our next solute. Notice how it is insoluble with all of the given solvents. This is due to the nature of the solvents, the nature of glycerol as a solute, the amount of solute, and the rate of solution. And with our final solute, we have palmitic acid and its solubility with the same solvents as the two prior solutes. Palmitic acid did not dissolve in water, dilute acid, dilute alkali, chloroform, hot alcohol, and carbon tetrachloride. However, it is soluble in ether and cold alcohol. This is because palmitic acid is highly soluble in organic solvents such as ether.
So, for our first question, explain the solubility of the different kinds of lipids used in each solvent. So, to answer to that, lipids are all insoluble in polar solvents like water, but highly soluble in the nonpolar or weakly polar organic solvents, including ether, chloroform, benzene, and acetone. In fact, these four solvents are often referred to as lipid solvents or fat solvents. So for our second question, did the above result confer with the expected result? Well, the answer is yes. It gladly benefited the expected result due to the reason that we are able to discuss and determine the principle in each of the tests employed for lipids. Also, we are able to determine what chemicals are used in each solvent, and we figured out the polarity of the liquids involved. Thank you.
So, the influence of bile on the action of lipase. The definition. Bile acids or bile salts are a complex digestive fluid produced by the liver. Lipase, specifically pancreatic lipase, is an enzyme that binds to the oil-water surface of fats and other lipids, where it then starts to absorb and digest the fats. It is important to note that lipases can only bind to and absorb the surface area of fats. Bile acids are emulsifiers, which help enlarge the surface area for lipase to act on. As for the chemicals used in this experiment, there is phenolphthalein, used as a laboratory reagent and pH value indicator. This means that its reaction depends on the pH value of what it's mixed with. There's also NaOH, otherwise known as sodium hydroxide, another reagent which is a corrosive chemical often used as a solid or 50% solution. The reactants used in this experiment are olive oil, neutral pancreatic extract, water, and bile. Here is a list of what each labeled test tube should contain. Make sure that the measurements of each substance are accurate so that the experiment can be replicated. And here is the procedure. Make sure to follow each step in order. First, shake all 5 test tubes thoroughly, then place the tubes inside an incubator for an hour. Once an hour has passed, take all the test tubes outside of the incubator. Place 1 drop of phenolphthalein in each test tube. Then titrate each test tube with 0.5 milliliters of sodium hydroxide until it turns pink in color. Finally, record the amount of sodium hydroxide used to neutralize the contents of each test tube.
So for this part, I'm going to discuss the result of the said experiment. So as what you can see in the picture, there are 5 test tubes with different colors. So sa first test tube, may content siya na 5 milliliters of neutral pancreatic extract. Tapos dinagdagan ng 4.5 milliliters of water plus olive oil. So, after siyang ilagay sa incubator for about an hour, ang naging color niya is red. So, meaning acidic siya. So, para just to neutralize the content itself in the test tube, kapag boiled siya, only 1 drop of sodium hydroxide. And if unboiled naman, pag unboiled, 2 drops ng sodium hydroxide just to neutralize the content in each test tube. Okay. So, nakadepende kasi yun sa color ng test tube kung acidic ba siya, neutral, or alkaline. So, makikita natin sa first test tube, color like dark red talaga siya. Meaning, super like battery acid talaga siya. Super acidic talaga siya. So, for the next one, medyo nag-light yung color. And umibabaw na rin yung olive oil. So, kaya ang nasa second test tube naman, ang content niya is 9.5 milliliters of water plus olive oil. So, alam naman natin na mas mabigat never naghahalo ang water at ang oil. Kaya meron kayo nakikitang oil sa ibabaw. Kasi yan yung olive oil. So, just to neutralize the second test tube, the content in the second test tube, we only need 1 drop of sodium hydroxide. So, for the third test tube naman, as what you can see, mas light siya than the last 2 test tube. Ang laman po niya is 8.5 milliliters of water plus meron siyang bile and olive oil. To neutralize the content in that test tube, we only need 1 drop of sodium hydroxide. So, kung makikita kung meron kayong copy ng pH scale, siguro nasa level 1. Yeah, level 1 to 0 to 1 pa rin siya. Acidic pa rin siya. Okay. For the fourth test tube, meron tayong nilagay na 5 milliliters of neutral pancreatic acid plus 3.5 milliliters of water plus 1 milliliter of bile plus olive oil. So, makikita natin na parang purple na siya. Purple to pink. And just to neutralize the content in that test tube, if boiled yung test tube nyo, we only need 1 drop of sodium hydroxide, and then for the unboiled one, we only also need 1 drop of sodium hydroxide. So, yung fourth test tube, makikita natin na ibang-iba yung color niya from the last 3 test tube. Parang purple na siya, parang level 12 na siya sa pH scale indicator, and it says here na alkaline na siya. So, ang progress kasi ng pH indicator is from orange, yellow to greens. Greens to blue is the neutral, and then parang blue to purple to dark purple is alkaline. So it shows here in the picture na alkaline na siya. And then last one, for the last test tube, we have 5 milliliters of neutral pancreatic extract plus 3.5 milliliters of water plus 1 milliliter of bile plus olive oil. So, yon. Just to neutralize the content itself in the last test tube, pag boiled, 1 drop ulit, and then kapag unboiled, 1 drop as well.
Hi, my name is Jason Marian D. Lalio, and for this part, I'm going to be discussing the questions and answers. Number 1, what is the role of bile acids in fat digestion and absorption? Bile acids has two following roles. One is emulsification of lipid aggregates. Bile acids have the effect of cleaning the particles of dietary fat, breaking down fat globules, and emulsifying them into small fine droplets. Emulsification is not digestion itself, but it is important because it significantly increases the surface area of fat and makes it available for digestion by lipase, which cannot enter the lipid droplets. Last role is solubilization and transport of lipids in an aqueous environment. Bile acids are lipid carriers and are able to solubilize many lipids by forming micelles, aggregates of lipids such as fatty acids, cholesterol, and monoglycerides that remain suspended in water. Bile acids are also critical for transport and absorption of the fat-soluble vitamins. Last question, what happens to fatty acid and glycerol once they have been absorbed in the digestive tract? They are transported through the lymphatic system and into the bloodstream in the subclavian vein below the clavicle.
What does olive oil represent in the experiment of lipids? Well, lipids are compounds that are insoluble in water but are soluble in organic solvents such as ether and chloroform. Lipids that are important to our discussion include fats and oils, fatty acids, phospholipids, and cholesterol. Fats and oils are esters of glycerol and 3 fatty acids. They are important in the diet as energy sources and as sources of essential fatty acids and fat-soluble vitamins, which tend to associate with fats. Most vegetable oils are weak acids. Since they are not soluble in water, their acidity cannot be measured in terms of pH levels. It is usually estimated in percent free acidity. Oleic acid, which is the major fatty acid in olive oils, is a monounsaturated omega-9 fatty acid. Monounsaturated fatty acids are a type of unsaturated fat. Mono, meaning one, signifies that monounsaturated fats have only one double bond. Owing to its beneficial effect on the muscles, tone, and activity of the gallbladder, olive oil stimulates the digestion of lipids because it is emulsified by the bile and it prevents the onset of gallstones. The bile dissolves the oil into the water contents like detergents that dissolve grease from a frying pan, splitting the oil into thousands of smaller droplets of oil. Because oil is not
insoluble in water, it is easy to see that the oil in the 4 test tubes is not mixed with other liquids.
So, phenolphthalein is an organic compound, the phenol family that is widely employed as an acid-base indicator. So, basically, the role of phenolphthalein is to indicate if the substance is a base or an acid. Naturally, colorless siya, pero nagiging pink siya kapag na-mix siya sa base solution. So, makikita natin sa structure na si acid and base are equilibrium to each other. So, ano may ibig sabihin na equilibrium to each other? So, if naglagay tayo ng hydrogen ion, which is found sa acid, sa pink solution, mag-switch siya into a colorless. But, if naglagay tayo ng hydrogen ion, which is found on base, magiging pink solution naman si phenolphthalein. So, makikita natin sa structure na si acid and base are equilibrium to each other. So, ano may ibig sabihin na equilibrium to each other? So, if naglagay tayo ng hydrogen ion, which is found sa acid, sa pink solution, mag-switch siya into a colorless. But, if naglagay tayo ng hydrogen ion, which is found on base, magiging pink solution naman si phenolphthalein.
Good day, I'm Janela May Labrador and my topic is about how is pancreatic lipase specific in regard to triglycerol. So, the pancreatic lipase and its protein cofactor colipase are required for efficient dietary triglyceride digestion. So, pancreatic lipase is usually secreted by the pancreas and transferred to the duodenum to participate in the hydrolysis and digestion of fat, cholesterol esters, and fat-soluble vitamins. So, si pancreatic lipase daw po, sinesecrete siya ni pancreas at transfer doon sa duodenum para makapag-participate po doon sa hydrolysis, digestion of fat and cholesterol esters, and fat-soluble vitamins. So, sa duodenum, siya yung first part of the small intestine, at yung main role niya is to complete the first phase of digestion. Triglycerides are the major dietary fats. They are hydrolyzed in the gut by lipases, the fatty acids, and monoglycerides. So, dietary fats are essential for life and good health. So, si pancreatic lipase po, in-hydrolyze niya si triglyceride to fatty acids and glycerol. So, when we say to hydrolyze a bond is to break it apart with water. And pag in-hydrolyze si triglyceride, it can produce glycerol and 3 fatty acids. So, triglycerides can be hydrolyzed to produce glycerol and 3 fatty acids in the presence of acid and heat or with a suitable lipase enzyme under biological conditions.
I'm Abigail Inubaya and I will be discussing with you about the function of bile acid, or what you call salt, under fat. Bile salts break down larger fat globules in food into small droplets of fat. Smaller fat droplets are easier for the digestive enzymes from the pancreas to process and break down. The bile salts also help the cells in the bowel to absorb these fat droplets. So, with that being said, bile acids are one of the primary components of bile. Bile is a greenish-yellow fluid made with the liver and stored in our gallbladder. It also helps with the digestion of fats in our bodies. They also help us absorb fat-soluble vitamins like A, D, and E, and K. In addition to that, bile contains cholesterol, water, bile acids, and pigment bilirubin. The role of it in the body is to aid digestion by breaking down fats, help absorb fat-soluble vitamins, eliminate waste products. Bile and bile salts are made in the liver, so it is stored in our gallbladder between meals. After we eat and there are fats present in our digestive tracts, our hormones send a signal to our gallbladders to release bile. The bile is released into the first part of our small intestine called the duodenum. This is where of the digestion happens. The bile helps to process and digest fats. Another primary function of bile is to remove toxins. Toxins are secreted into the bile and eliminated in feces. A lack of bile salts can cause a buildup of toxins in our body. That's all. Thank you.
Hello everyone, I am Ilyana Mystica and I will be reporting on behalf of Miss Audentia. I will be using her script to explain her part. So let us start. What is micelles of lipid digestion? Micelles are non-permanent molecules that arises during the digestion and absorption of fat. Micelles are water-soluble and allow for the passage of lipid digestion products to the small intestine surface for absorption. Cholesterol and fat-soluble vitamins are also included in the core. Lipids would sit on the surface of chyme without micelles, never coming into contact with the epithelial cell's absorptive surfaces. Micelles can readily fit between microvilli and approach quite close to the surface of the luminal cell. Lipid compounds exit the micelle at this moment and are absorbed through simple diffusion. Micelles in the body allows for the absorption of molecules that are insoluble in water, such as lipids. They also carry lipid-soluble vitamins to the small intestine. So what does happen with them in the epithelial cells of intestine? Micelles can easily squeeze between microvilli and get very near to the luminal cell surface. At this point, lipid substances exit the micelle and are absorbed via simple diffusion. On the other hand, triglycerides are formed from free fatty acids and monoxyl glycerides that enter the epithelial cells. A protein sheath surrounds the triglycerides, which are combined with phospholipids and cholesterol. This new compound is a water-soluble lipoprotein known as chylomicron. Chylomicrons are discharged from the cell after being processed by the Golgi apparatus. Chylomicrons are too large to pass through the basement membranes of blood capillaries, thus they enter the huge pores of lacteals instead. The lacteals come together to form the lymphatic vessels. The chylomicrons are transported in the lymphatic vessels and empty through the thoracic duct into the subclavian vein of the circulatory system. Once in the bloodstream, the enzyme lipoprotein lipase breaks down the triglycerides of the chylomicrons into free fatty acids and glycerol. That is the end of our report, and we hope that you learned something from us. The next slide contains the references that was used throughout the report.